In our previous session, we discussed the molecule activity predictor, the keep, and the trim functions. So the molecule activity predictor basically uses the ingenuity data to increase or decrease the measurements of certain molecules, as an example, morphine, and to see what effect that has on predicted activation or predicted inhibition of upstream or downstream molecules. We also discussed the keep function, which allows us to remove certain relationships, to keep certain relationships based off our certain set specifications using the filter options. So for example, if you want publications from a certain date range, or if you want certain cell lines, we can use the keep function to only have those certain relationships in our pathway. And we also discussed the trim function as well, which allows us to remove certain relationships based off our set specifications using the filter tab, right? So if you want to remove, certain cell lines, or if you want to, remove a certain publication date ranges, right? So we can use the trim function to do that, right? After we've already created a pathway. In today's session, we're just going to be reviewing all of our learned concepts from all our previous sessions, right? And we will also be having our quiz at the end of today's session as well. So the quiz is a 40-question quiz. And, after our review session and our question answer session, we will be having the quiz. So as I mentioned prior as well, there is accumulated points from our previous sessions that will be going towards this final quiz. So we'll just be going over the points that have been added throughout all our previous 4 sessions. So coming in first place, whoever came in first place for any quiz would have received an additional 1,000 points. Second place, 800 points. Third place, 600 points. Fourth place, 400. And fifth place, 200. This has already been uploaded to the Google Drive, so you can go ahead and look at this. So these have been all of the winners throughout all of our prior quizzes. So you can just go ahead and look at that as well on the Google Drive. So this is the accumulated points after all the 4 sessions. So you can go ahead and look at this, but this will be accumulated after our quiz today. So these points will be added to the final score of today's session of today's quiz. Right. So we'll get into today's session now. Prior to doing that, we'll just review the tasks from our last session. So in our last session, I asked you to connect midazolam and ethanol to GABA, and I want you to include the shortest paths plus 1 in your pathway. And I want you to then use molecular activity predictor to increase measurements of both midazolam and ethanol, right? So you can see here that midazolam and ethanol have increased measurements. They turn red, right? And you can see that it has a downstream effect on GABA by predicted inhibition of GABA down here, right? So it is a slightly lesser shade of right? So it has a less confidence in the predicted inhibition, right? Then conversely, part of that task, I also ask you to decrease the measurements of midazolam and ethanol. So by decreasing the measurements, you see that the colors have both turned green here, right? So midazolam and ethanol are both green here. And you see that it has a downstream effect of increased activation, of predicted activation rather, of GABA. It is a lighter shade of orange right here, so that means that it has less confidence in the predicted activation, but it is predicted activation nonetheless. So like we said, today will be a review session, so we'll be going over the learned concepts from all of our previous sessions. We'll have a question answer session after that if anyone has any questions regarding any of the concepts. And then after that, we will have our final quiz, which is a 40-question quiz. All right. So we first started off our session, our workshop by going over the research hierarchy, right? So at the bottom of our research hierarchy, if you recall, we said that there was editorials and expert opinions. So the reason that editorials and expert opinions are at the bottom of our research hierarchy is that they are not founded by any empirical evidence. They're just theory-based. They are the opinions of experts in a certain field, such as researchers or physicians, right? And it is their professional, opinions that determine editorials and expert opinions. They're not founded on any sort of empirical data being observational data or experimental data. So it has no empirical data in it. That's why it is at the bottom of our research hierarchy, right? Then above editorials and expert opinions, we discussed like last time as well, there is in silico analysis. And the reason that in silico analysis is below all these other research types is because it is not any sort of biological process that's actually occurring. It's just using computer simulations that utilizes our current understanding of biological processes to make a prediction on how things will look, but not actually conducting that biological process. It is just predictions of how it might look, right? That's why it is below all these other ones. Above that, we have our case reports, cross-sectional studies, case control studies, cohort studies. These are our different types of observational studies. So case reports is the least significant, has the least significance of our observational studies because it only involves one participant. All the other observational studies, so cohort, case control, cross-sectional, they have multiple participants. That is why they have greater significance, greater quality, as opposed to case reports, right? And above our observational studies, we have randomized control trials, which is an example of an experimental study. Then at the top, we have systematic reviews and meta-analysis. So these are our highest significance, highest quality studies. And the reason for that is that because they are an accumulation of the results, a culmination of the results of our different observational and experimental studies. So randomized control trials, cohort, case control, et cetera, right? And all of these have some sort of impact on healthcare systems, right? So we also discussed in our first session that there's two basic classifications for research, right? That we can basically divide researches on, those being observational studies and experimental studies. So observational studies, these are basically studies where you do not make any sort of intervention. You let things go in its natural course. So say, for example, we're doing an observational study on diabetes. You do not make any intervention, to diabetes. Just observe, okay, do participants develop diabetes? What complications develop from diabetes? No intervention is made. And if you're looking at diabetes example as well for experimental studies, it would be like in this case making some sort of a medication for diabetes or a surgical technique, right? So making some sort of intervention is experimental. Just observing without making any interventions, that is observational. So the examples of observationals that we discussed were cohort, case control, cross-sectional case reports. And examples of experimental studies that we discussed in our first session were randomized control trials and crossover studies. Okay. And then for the major types of observational studies, those being case control, cross-sectional, cohort, the way that I had you understand these was that, okay, think of case control studies as sort of the past, okay? So if we're looking at the example of diabetes again here, right? These would be collecting participants that already have diabetes and participants that don't have diabetes and seeing, okay, what led the participants that have diabetes to get it in the first place, right? So what were the risk factors that led them to develop diabetes, right? So that is already the past. You have accumulated participants that already have it, right? And the statistical method that is used to calculate risk or determine risk is called odds ratio. So case control studies deal with odds ratio. That is something to note, okay? Case control studies, just know that case control studies are associated with odds ratio. Then cross-sectional studies, you can sort of think of it as the present. So cross-sectional studies are used to determine often the prevalence of a certain condition or a certain disease, etc., whatever you're analyzing, right? So it is your classic surveys or questionnaires. So with our example with diabetes, again, we can think of it as, okay, asking participants, however many of them have diabetes, and say after your survey, you find that 3% of the population has diabetes, that would tell you the prevalence of the certain condition or whatever you're analyzing, right? So it is the present condition of however it looks. Our cohort studies are the future. So in this, you collect participants that have certain risk factors. So with our example with diabetes, would be collecting participants that have, say, for example, a poor diet, sedentary lifestyle, are overweight, right? So these risk factors and seeing, okay, what percentage of those participants eventually develop the condition that you want to analyze. So in this case, diabetes, with those risk factors, how many of them develop diabetes? And you also look at participants that don't have those risk factors. So is looking at the future, you do not have any cases yet, but you are seeing how many of those participants end up becoming cases. And the statistical method that is used to calculate risk for cohort studies is relative risk. So case control, know that it is odds ratio. Cohort studies, know that the statistical test that's used is relative risk. That is important to know, okay? And then we also discussed the experimental model that is used often for the drug design process, right? So at the bottom of our pyramid, we have computational, so in silico studies. So in silico basically means within silicon, so using computer studies to simulate certain biological processes, right? So that's, again, like the software that we use, IPA, right? It could be the utilization of IPA or other in silico techniques, right? So that would be our first step is the drug design process, and that comes before any of the other steps, right? In our drug screening process, what we do is that we utilize in vitro studies. So in vitro in Latin means within glass, right? So this would be your classic petri dish studies or test tube studies, right? So it is your sort of laboratory studies, right? And it's your cell culture studies is part of your drug screening process, right? After that, you move on to your preclinical stage, which would be your animal studies, your in vivo studies. So in Latin, in vivo means within the living. So you do this prior to moving on to your clinical studies, which are in humans, which occur after you've already conducted your in vitro studies, in vivo studies, or in silico studies as well, right? So human studies, clinical studies come at the end after you've conducted your animal studies, cell culture, your in silico analysis. And we also discussed that systematic reviews and meta-analysis were the most significant and the highest quality of research, right? So both of these are quite similar, right? But there are some differences. So both of them involve some sort of statistical method of collecting data. They have inclusion, exclusion criteria. You don't just include all of the data from your research collection, right? You include certain criteria. You include certain, certain data, you exclude certain data based off your specification. So your method section includes which data you've included, whether it be based off of the most recent literature or analyzing a specific cell line, right? You make sure that you include that in your method section, and both of them have that systematic way of collecting data, right? So systematic reviews, right? They have a systematic method to obtaining to answering a specific question, and oftentimes they generate a new hypothesis as well, right? So they end up creating new questions by the end of it, right? Just know that oftentimes it generates hypotheses as well with the process of going through a systematic review, and systematic reviews can become meta-analysis, right? So the basic difference here is that like meta-analysis, they have some sort of statistical processing of data, right? So generate some sort of statistical data such as p-values, z-scores. So any time where you generate statistical data, that is a meta-analysis, right? And systematic reviews, if they have some similar studies together, right, it can maybe generate statistical data. In that case, systematic reviews can become meta-analysis, right? But note that for meta-analysis, there is the generation of statistical data, which differentiates it from systematic reviews. And what you can also do with systematic reviews is that because you are accumulating multiple different studies, what that does, it increases the power of the study. So the power is the likelihood of your results, your statistical analysis to actually reflect reality, right? And how meta-analysis increases power is by increasing the number of participants or the number of participants that are in these studies. So meta-analysis, because you're accumulating similar studies. So say in one cohort study, there were 350 participants. In another study, there were 350 participants. In another, there were 200 participants, right? So what meta-analysis does is that it adds a 350 plus 350 plus 200 to have a total of 900. So because of that greater number of participants, it increases the power of the overall study. And that is another, utilization, another big advantage of meta-analysis because it mitigates the need for large clinical trials, right? We also discussed the drug design process as well. So at the beginning of our drug design process, it is our basic research stage. So like, say for example, we're looking at diabetes. That's the example that we used last time as well. So in our basic research stage, you might have a 20,000 compounds that we identify that have some sort of effect on diabetes, right? So that would be our basic research stage, right? We identify more than 20,000 different compounds that have some sort of relationship to diabetes, right? Then in our stage 2 process, right, this would be our discovery of a certain target. So for example, we want to look for a specific target of those diabetes, compounds that we've identified. So in this case, say for example, we want to select for compounds that are secretagogues. So secretagogues are diabetes drugs that increase the release of insulin. So you want to select for secretagogue specifically, just as an example, right? So upon selecting for that specific target, we might find that there are 5,000 to 20,000 different compounds that we've identified. Stage 3 is our preclinical research. So this would be our animal studies. So in our stage 3 process, our preclinical research stage, we might find that there's 250 compounds that we've identified. Then stage 4 is our clinical research stage. This would be our human studies. We might find that there are 5 compounds that we have identified, moving from greater than 20,000 to 5 here. Then finally, our stage 5 is our drug approval process. This is before we approve it to the FDA. So the FDA might just end up approving just one compound. So in our basic research stage, we might have identified 20,000 compounds, right? By the end, like when we approve our drug by the FDA, there might just be one that ends up getting approved, right? So that's just the whole like process, how it goes, right? So our utilization of in silico analysis and silico techniques come in the stage 1 and the stage 2 process, our basic research and the discovery of drug target. So normally this might take, without the utilization of in silico techniques, this could end up taking years or months to years, right? But with the utilization of in silico techniques, this is going to be drastically reduced to weeks, right? So it drastically reduces the time it takes in stage 1 and 2 in the overall approval of drugs. So the software that we went over for the majority of our session was IPA by Kaizen, right? And the specific functions that we learned about were the grow function, the path explorer function, molecule activity predictor, keep, and trim. So I'll just be going over these different functions briefly. So just as a refresher for all of you all, so that everyone is aware of what the different functions are, right? So we began by learning about the grow function, right? So we learned that the grow function allows us to visualize all of the related molecules to whichever node that you're analyzing. So for example, you're analyzing morphine or buprenorphine. So it will show you all of the related molecules, whatever relationships there are with that specific molecule, be that morphine, buprenorphine, etc. It could be any sort of molecule, any sort of node as well, right? So show you all the related molecules that are upstream and downstream of that node, right? And it shows you indirect, direct, chemical interactions, activation, inhibition relationships, etc. Right? So these specific lines that connect the different nodes, those indicate the type of interaction, right? So you might have noticed that some lines are an arrow, some are like sort of a line that ends in sort of a snub, right? So depending on the shape of the line that's connecting two nodes together, that denotes the type of interaction. So it shows you direct, indirect interactions, chemical interactions, all this sort, right? So all that is shown with the grow function. So we'll just show how to actually use the grow function, how you would find the grow function. So we'll use our example with buprenorphine. So you would search for buprenorphine here. You can then click enter on your computer, or you can select it over here if it shows up. In this case, we'll just click enter on our computer. I'll be taken to this page right here. You can then click on buprenorphine right here. We'll add it to our pathway. We'll add it as a symbol, click OK, right? Buprenorphine gets added to our pathway. We then click on the build function right here, right? We can click select a tool. We can click on grow, right? It takes us to this page right here. I've already added it to our pathway, but this page basically allows us to filter for our searches, like I mentioned in our previous sessions. So say for example, you want to include direct interactions, you want to include indirect interactions, however many pathways you want or relationships you want to include in your pathway, you can add that over here. Whether or not you want to include upstream or downstream molecules, and how you want to sort your data as well, be that by the species, so human studies or animal studies, the tissues and cell lines, macrophages, nervous system, etc. Right? MicroRNA confidence level, all the different filter options, right? You can go ahead and do that here. And then you can click apply. In this case, I've just added all of the different pathways that I have not filtered for it whatsoever. And I get this right here, okay? Then you are also able to individually rearrange the different nodes, right? So you're able to move the nodes around, or you can also click over here, right? This allows you to see the different layout options that IPA provides you, such as subcellular, hierarchical, right? We did discuss that in a previous session as well. So you can move the nodes around, or you can use one of the layout options that IPA provides. In this case, I've just moved around myself, okay? And we also discussed the different nodes. So depending on the shape of the node, it denotes a different sort of thing. So like, for example, a cross like this denotes a disease. A drug denotes what's it called, an oval shape like this, right? So depending on the shape, it denotes a different sort of node, okay? These are all the different nodes that are provided for us by IPA, but there are certain ones that I want you to focus on, certain ones that you might find you deal with more often than others, right? So the ones just to keep in mind are chemicals and drugs, which are this oval shape like this, right? So a chemical or drug will appear like this, a disease will appear like this, a cross shape, an enzyme will be a diamond shape like this, a function will be an octagon like this, and a cytokine will be a square like this, right? So these are just some common ones that you will more likely than not come across, okay? And then we also discussed the different types of relationships, right? So depending on the type of line that is connecting it, it denotes a different type of relationship. So these are all the ones that are provided for us by IPA. So I have included this in the presentation. You can have a look at it later as well, but we'll just talk about the major sort of relationships or interactions that you will come across. One being direct interaction. So a direct interaction will be a solid line like this, right? If it's a direct interaction, it will be a solid line. If it is a indirect interaction, it will be a dotted line like this. If it is an activation relation, right, it will be an arrow, so it ends in an arrow like this. Or if it is an inhibition relation, it will end in sort of like a snub like this, right? So ends sort of like this, a line that's like this. Okay. So these are the ones that you will most likely come across, you most commonly come across. Right. We also discussed the path explorer function as well. So the path explorer function basically allows us to connect two or more molecules / nodes to one another and determine what relationships they have. So all the intermediary nodes between those two molecules / nodes, the path explorer function shows that, right? So say, for example, just as an example, you're connecting morphine with opioid receptor, right? So all the related molecules / nodes, right? It'll show that, right? Like, like say for morphine, there is a second messenger molecule. Then after that second messenger molecule, there might be a chemical that comes after that, right? Then you get to opioid receptor, just as a rudimentary example, right? So that will be shown with the path explorer. And much like the grow function, it shows you direct, indirect, chemical interactions, activation, and inhibition relations. So the specific lines that are connecting it, like these, right? It'll show you like, what type of interaction is connecting the nodes by, right? By the type of line that it is, right? That is connecting those two different nodes. So that is also shown. We'll just go over how to use the path explorer function. So continuing with our example with buprenorphine, we type in buprenorphine over here, right? We can then select for buprenorphine, shows up, or we can click enter on our computer. In this case, we just clicked enter. We can select for buprenorphine. We can add it to our pathway. We'll add it as a symbol in this case, right? And we'll click OK. It gets added to our pathway. Then to get to Path Explorer, we go on, or sorry, we'll add apoptosis as well, right? Because we want to compare buprenorphine to apoptosis. So we'll type in apoptosis in our search bar right here. We'll select it. We can either select apoptosis if it shows up, or we can click enter on our computer. In this case, we just click enter. We'll select the first one right here. We'll add it to our pathway. We'll add it as a disease or function. Then we'll click OK. So we have both of these in our pathway now, right? Then to get to the Path Explorer, we click on Build, we click Select a Tool, we click Path Explorer. We get taken to this page right here. So in this case, we'll add buprenorphine to set A. So we click on buprenorphine, it gets selected for. When it gets selected for, we know it's been selected for if it turns to a sort of blue color like this. So if it has a blue highlight, we know it's been selected for. We can then click Add. Buprenorphine gets added to set A. We can then do the same thing for apoptosis. So we can click on apoptosis, it gets selected for. We can add it to set B. Then we can also set the direction. So going from set A, which is buprenorphine, to set B, which is apoptosis. Going from set B, which is apoptosis, to set A, which is buprenorphine, or going from set B and set A at the same time, right? So any direction. So we can include whichever direction we want, but in this case, we'll be going from set A to set B. So buprenorphine to apoptosis, right? So once we've selected for that, which direction we want, and we have added whichever molecules you want in set A or set B, we can then click apply. And just as a reminder, you can also have more than just one or two molecules in set A or set B. So you're able to have multiple different molecules in set A or set B, right? But in this case, we just have one in each. Click apply, right? We get all the different relationships or pathways that have been found between buprenorphine to apoptosis. We can click on the individual relationships, or we can click here to add all of them at once. So we'll just click here to add all of them at once, and we'll add that to our pathway right here, right? Just add it to our pathway like that, and we have all the relationships. Then we can just go ahead and rearrange it to look somewhat more visually appealing like that, okay? Then we also discussed how to filter our searches, right? So over here, we have our different filter options. We have our data sources, microRNA contents level, our species, right? For the majority of these examples, we did not filter, but just know that you are able to filter. And the filter options you have available to you are the data sources. So whether that is the Ingenuity data that you want to include, third-party sources, that being the mouse genome database, microRNA, mRNA interactions, you have multiple different, third-party sources that are available to you. The confidence level, whether or not it is experimentally sounded or not, the species, so human studies, or animal studies, tissues and cell lines, so macrophages, nervous system, liver, etc. Right? Whichever tissue or cell line you want to select for, mutations. So whether or not that relation leads to a certain type of mutation, such as a wild type mutation, a translocation, etc. Right? There's multiple different mutation options that are available to you. The type of relationship type, so whether it be an activation relation, inhibition relation, right? You're able to select for that as well. Publication date range, whichever publication date range you want to include into your data. The node types, the diseases, whichever nodes have a relation to a certain disease, and biofluids as well. So which nodes are found in which biofluids, you're also able to include for that, right? So you have all these different node options that you're able, or filter options rather, that you're able to select for. And the significance of these different filter options is that oftentimes, it may be required as your inclusion or exclusion criteria for your research. So we discussed that in systematic reviews and meta-analysis, there are often, inclusion, exclusion criterias. You don't just include all of the, data that you find, right? You want to, basically select for specific data, right? So it has utilization with systematic reviews and meta-analysis, right? And oftentimes, you might find that prior to filtering, there are a large number of connections, but after filtering, that number gets drastically reduced. So that is another benefit of filtering, right? It reduces the number of overall relationships or connections that you have. And you often find that there is a different target effect after you filter the data. So prior to filtering, you might find that one compound might be inhibited, right? But after you filter, you might find that that same compound is now activated, right? So it is a different effect based off of your filtering that you've done, right? So that is another utilization for it. So the example that we looked at how it might look after filtering was with morphine. So morphine over here, this is prior to filtering, we found that there is a total of 307 relationships, right? And after filtering for macrophages and publications between 2000 to 2024, we found that there were a total of 23 relations. That is a drastic reduction in the total number of relationships. But just note that there may not always be a reduction in the total number of relationships. From my own personal experience. So after filtering from my personal experience, right, I found that with one specific case, there was actually an increase in the number of relationships, and that has something to do with the algorithm of IPA, right? But just note that for the majority, right, the total number of relationships do decrease after you filter, right? We also discussed the different layout options that IPA provides for you, that being the organic, circular, radial, hierarchical, subcellular, different layout types, right? To get to the different layout options, you click over here, right? That takes you to this tab right here, and you're able to select for the different layout options. So if you selected for organic layout, it looks something like this. If you select for a circular layout, it looks something like this. So all the different nodes are on the periphery. And you're able to look at the individual lines that connect the different nodes. So that is visible for you. If you select for a radial layout, it looks something like this. So you have the central node in the center, right? And all the other nodes on the periphery. If you select for a hierarchical layout, it looks something like this, right? So, again, I included this in our in this example because this is quite literally what IPA showed for me, right? The nodes are quite spread apart, right? But I just included just to show that sometimes IPA might show you a layout that looks quite impractical because the nodes are so far apart. Just note that you can always move around these nodes to fit your research needs or to fit the visual needs as you see fit, right? So you're also able to move the nodes. But an example of how a hierarchical layout would look without the nodes being spread apart is like this, right? So sometimes it may show the pathway looking like this. In other cases, the nodes may be spread apart like this. So just know that you can change it to your own needs. You don't have to just rely specifically on what IPA provides for you, right? And then we also discussed the shortest paths and the shortest paths plus one. So the shortest paths and shortest paths plus one is a part of the Path Explorer. So when you're connecting one or more nodes slash molecules to another set of nodes or molecules, right? It basically shows the shortest paths right here, or the shortest paths plus one. And it basically what this means is that, okay, the number of intermediary nodes that is able to connect those two molecules together by, right? The shortest path will show you the least number of nodes that you can connect those two molecules by, right? So in this case, we connected ketamine and morphine to NMDA, NMDA being a neurotransmitter. After we connected ketamine and morphine to NMDA, the shortest paths found that there were two intermediary nodes that could connect ketamine and morphine to NMDA, right? And the shortest path plus one is just one additional node to the shortest paths, right? So if the shortest path is two nodes in this case, the shortest path plus one will be three nodes that can connect, in this case, morphine and ketamine to NMDA, right? But just note that it doesn't necessarily always have to be two for the shortest paths and three for the shortest paths plus one, right? It can also be one for the shortest paths, one intermediate node for the shortest paths, and two intermediate nodes for the shortest paths plus one, or it could be three intermediate nodes for shortest paths and four for the shortest paths plus one, right? So just note that shortest paths is always just one additional, and shortest path is just the least number of nodes that can connect it, right? That's the difference. And the sort of example that I use to demonstrate the shortest paths and shortest paths was with morphine and ketamine connected to IL-1. So if we connected morphine and ketamine to IL-1, selected specifically for the nervous system and activation, causation, correlation, and expression relationship types, we found that there was only one interaction, one relationship that could connect it, and that was only with morphine. Ketamine did not have any sort of interactions with IL-1, right? So that's what we found with just the shortest paths, right? But if you included the shortest paths plus one, which is a total of 10 relationships, right? We found that, in fact, ketamine does have an interaction with IL-1, but that was not shown with the shortest paths, right? So just, that's another thing that you can look through with your research, just to check both the shortest paths and the shortest paths plus one to see if there are any interactions that are, being shown with either or, right? We also discussed the molecule activity predictor. And basically what the molecule activity predictor does is that it uses the ingenuity data to see that, okay, say you were to increase the measurement of a certain molecule or a node, right? Or you were to decrease the measurement of a certain molecule or node and see, okay, what effect that has on the upstream or downstream molecules in their predicted activation or their predicted inhibition, right? So it uses the ingenuity data, the IPA's own ingenuity data and third-party sources as well to see that, okay, based off of our data, right, that from the ingenuity data, if you were to increase the measurement, what effect that would have on downstream or upstream molecules. So different colors denote different things. So if it's a red color like this, it means that there's increased measurements of that certain molecule, right? If it is a green color like this, that means there is decreased measurements of that certain molecule. If it is predicted activation, it will be an orange color like this. If there is predicted inhibition, it will be a blue color like this, right? And the degree of saturation of that color. So if it is a darker blue or a darker orange versus a lighter blue or a lighter orange, that denotes the level of confidence in the predicted activation or inhibition, right? The different lines that connect the different nodes by, right? So the different relationships, right? The color of that line denotes a certain thing, right? So if it is a leading to activation, right, then the line will appear as a orange line, right? If that will be leading to an inhibition, if you increase or decrease the measurements, right? If it appears as a blue, that means that it's leading to inhibition, right? If it appears as yellow like this, after increasing or decreasing measurements of a certain molecule, that means that the findings are inconsistent with the state of the downstream molecule. And after increasing or decreasing the measurements of a certain molecule, you find that there is a gray line, that means that there is no predicted effect using the ingenuity data, right? So that is basically, the color coding, right? The color scheme that is used with the molecular activity predictor. So we'll be going over an example of how to use molecule activity predictor as well. So this was an example from last time, right? So we'll type in ethanol. We can then either select ethanol over here if it shows up, or we can just click enter on our computer. In this case, we'll just click enter. We get taken to this page right here. We can click on ethanol here. We can add it to our pathway. In this case, we'll add it as a symbol, right? Then we'll click OK. It gets added like this. Then we can go ahead and add TFEB as well, right? So we're connecting ethanol to TFEB. So we'll type in TFEB right here. If it shows up, we can just click it here, or we can just click enter on our computer. We'll just click enter. We get taken to this page right here. We'll click on the first one, TFEB right here. We'll add it to our pathway. We'll add it to new my pathway one, which is this pathway that is open right here. Okay. We'll add that. Then we have our two different nodes entered to our pathway. Then before we get to molecular activity predictor, we have to connect both ethanol and TFEB together. So we'll go to the build function right here. We'll click select a tool. We'll go on Path Explorer, right? Get taken to this page right here. We'll select for ethanol. We make sure that ethanol is selected for it has a blue highlight. We'll click add to set A, right? And we'll make sure that our direction is going from set A to set B, so ethanol to TFEB, right? We'll also add TFEB. We'll select for it. It turns blue highlighted like this. We'll add it to set B, add right here. So what's called ethanol and TFEB are both set as set A and set B, right? We'll then also specify specifically for macrophages. So that was the specification that we made in our prior example in our last session, right? So we'll specify for macrophages. Then we'll go ahead and apply those changes, right? Then we see that there is only one interaction that was found between ethanol and TFEB. We'll click here and we'll add it to our pathway. We get this right here, right? Then we'll just go ahead and make the pathway somewhat more congruent. We'll just make it like somewhat more visually appealing like this. We just rearrange it, moving the nodes around. Then to get to molecular activity predictor, what we do is that we click on overlay right here, right? So this button, click on overlay. We click on select a tool. We go to molecular activity predictor right here. That takes us to this page right here. So just make sure that your prediction is on prior to actually using molecular activity predictor, because sometimes you might find that prediction is off and you increase the measurements or you decrease the measurements of a certain molecule and nothing happens, right? So just make sure that prediction is on. It says predicted on like this. You can set the direction upstream or downstream, right? Showing the predictions for upstream or showing the predictions for downstream. And you can use these buttons right here. So this red button right here is for increased measurements of a certain molecule. So if you click on this, you will notice that your cursor changes to a paint icon, right? So that tells you that if you click on a molecule, that molecule will then show increased measurements, right? And this green icon right here is for decreased measurements, right? So like I said over here, you can set the direction going from upstream only, so showing the predictions for upstream molecules only, showing the predictions for downstream molecules only, or upstream and downstream at the same time. In this case, we'll just go for downstream, okay? So we'll select for that. And then we'll select for increased measurements. We make sure that our prediction is on as well. So we click on here. Then we can click on ethanol. We see that ethanol turns a red color like this. And since this was a prior example, you would remember that ethanol had no connections with, had no sort of predicted activation or predicted inhibition with just the shortest paths, right? So this is what we found. There was no prediction of activation or predicted inhibition, right? The same thing was true for the decreased measurements. So we clicked on decreased measurements. We saw that our cursor changed to a paint icon, right? Then we clicked on ethanol. Ethanol turns green like this. We saw that there's no predicted activation or inhibition of any downstream molecules. However, if we added the shortest path plus one, right? We click on shortest path plus one. We go to shortest path, click on shortest path plus one. We add these specific interactions to our pathway, right? We see that, in fact, there is a predicted activation of TFEB, albeit with less confidence, right? But there is nonetheless a predicted activation if we increase the measurements of ethanol, right? And the opposite is true if we decrease the measurements of ethanol. If you decrease the measurements, there is a predicted inhibition of TFEB, albeit with less confidence, but nonetheless, there is a predicted inhibition, right? Then finally, we also discussed the trim function and the key function. So the trim function basically allows us to remove certain relationships in our pathway based off our set specifications, right? So using this filter option right here, right? The same filter option that we had for the grow function or the path explorer function. If you already have a pathway created, right, you can use the trim function to remove certain relationships based off set specifications, right? And the example that we used in this case was of morphine. So we'll continue with that example, right? So, I've already gone ahead and added morphine to our pathway. I've used the grow function. I have not filtered for it whatsoever. And we see that there are a total of 307 nodes and 307 relationships that were added, okay? Now to get to the trim function, we click on build right here. We click on select a tool, and we click on trim. That takes us to our filter options, right? And in this case, I have filtered for ingenuity, expert information, and human studies. So I do not want human studies and I do not want ingenuity, expert information. I only want the third-party information right here, right? And human studies and ingenuity, expert information will be removed, okay? So we can go ahead and apply those changes, right? And we see that this is remaining, right? So these are the relationships that are remaining, and 265 rather nodes were removed, and 265 relationships were removed as well. So the nodes being these individual points right here, the relationships being the lines that are connecting them, right? So 265 nodes and 265 relationships removed. Conversely, we have the keep function. So if the trim function removes relationships, the keep function keeps those relationships based off your set specifications using the filter tab right here. So, say you want to filter for, the type of data source you want, the publication date range, right? You're able to do that with the key function. And the example that we used last time as well was with morphine. So we'll go ahead and do that, right? So, again, we have morphine added. We use the grow function to grow morphine. There was no filters applied here, right? So there are a total of 307 nodes and 307 relationships, right? You can then go on the build function right here. We click select a tool. You can click on keep, right? That takes us to our filter options right here, right? So we can filter for our options as we see fit. In this case, we'll keep it, continuous with our example here, where we filtered for ingenuity, expert information, and human studies. So this removes ingenuity, expert information, and human studies. In this case, we'll be keeping this. So we'll be keeping ingenuity, expert information, and we'll be also keeping human studies, right? So we'll see what effect that has on the pathway, right? So we go ahead and apply those changes. And we get this right here, right? So 265 nodes were kept, right? And 0 relationships were kept. So, these nodes did have some sort of relationship with human studies and the ingenuity expert information, but no relationships between these nodes were found with morphine, however, right? So these nodes did have some sort of interaction or some sort of relationship in human studies and ingenuity expert information, but no relationships between these nodes were found with morphine. That's basically what that means, right? So that is the end of our review session, right? That was all the major topics that we discussed throughout our sessions. If anyone has any questions, I can answer any questions that anyone has before we move on to our quiz.
A question that I was attended each and every quiz, but I couldn't get distinction. So will my marks count? Like, will my point count in this quiz?
Yes. Your points will count. Whatever points you get in this quiz will be as they are. But if you came in first, second, third, fourth, or fifth in any of the prior quizzes, then just some additional points will be added. So I'll just be going over what points will be added. So if you came in first for any of the quizzes, 1,000 points will be added. Second, 800 points will be added. Third, 600. Fourth, 400. Fifth, 200. Right. So those points will be added, but you will still be getting the points from this quiz, right? Just additional points will be added if you had first, second, third, fourth, or fifth.
So may I
Sure.
ask,
Yeah.
sir, you told us about functions of IPA, where you told grow function and path explorer. Sir, I want to know about the molecule activity.
Yeah, so molecular activity predictor, basically what that does is that, okay, you can use the grow function or the path explorer function as well to then upon use molecular activity predictor, right? And what molecular activity predictor does is that it uses the data that IPA has accumulated basically from all of the different literature that, IPA has at its disposal, right? And it uses that literature to see that, okay, say you were to increase the measurement of, let's say, morphine, right? So you use the grow function or use the path explorer function, right, to connect morphine to another molecule, or you just use the grow function to grow out morphine, right? So you have your pathway right here, right? And what the molecular activity predictor allows you to do is that, okay, say you have a lab setting, right? In the lab setting, you are increasing the measurements of morphine, right? So what that does is that it simulates that sort of lab environment that by increasing morphine, what effect does that have on the upstream or downstream molecules to morphine, right? In a certain biological pathway, right? So that's what the molecular activity predictor does, is that it simulates that biological environment using our data from all the different literature that it has, right? And it uses that literature data to come up with predictions of how it might look. Basically, that's what the molecular activity predictor is.
Okay, sir.
Including the QR code for the quiz. So, sorry, not this page right here. So you can go ahead and, scan the QR code on your phones. And upon scanning the QR code, you will be taken to this page right here where you have to enter the game pin, right? So you can just take some time to scan the QR code, then I will show you the game pin after as well. The game pin for the quiz is so the game pin is right here. I'll also share it onto the chat as well. Okay, so you can also click on the game pin right here to get to the quiz, or you can also just type it in from the page right here. We'll give until around, let's say, 11:56 for everyone to join. And if anyone needs any additional time, we can do that as needed. But we'll give until around 11:56 to start the quiz.
11:56.
The duration will be approximately around half an hour. So there are a total of 40 questions. It should take around half an hour to complete.
Okay, sir.
Okay, has everyone been able to join the quiz to enter the game pin in? So let's check the chat. Okay, does anyone need any more additional time, or can I begin? Okay, so we'll be beginning now. So I'll be starting the quiz now. Okay. There are a total of 40 questions, by the way. Okay, so our first question is, what is the observational study that has the least significance? And the answer was case reports. If you remember, I mentioned that since case reports are only one participants, they have the least significance. Which of the following statements regarding systematic review is true? Right. So the answer was the results from the study must be reproducible. Which function of IPA allows one to visualize all the related molecules to know that they are analyzing? And yeah, this was the grow function. Grow function allows you to show all the related molecules to the one that you're analyzing. So for example, morphine, you grow morphine, shows you all the related molecules. All right. What does the green color in molecule activity predictor denote? So this green color right here, what does that mean? And the answer was decreased measurements. So the green color denotes decreased measurements of that certain molecule. What is a commonality between systematic reviews and meta-analysis? All right, so the answer is that they both follow systematic method to collection of data. Only meta-analysis generates z-scores or p-scores, and this is false. They both do follow a systematic method. All right, which of these is an example of an experimental study? All right, so the example was randomized control trials. All these other ones are observational studies. What does the symbol below represent in a pathway? So this symbol right here. Yep, so the answer was a drug or a chemical. It can either represent a drug or it could also represent a chemical as well. Which of these most accurately describes what in vivo means? Right. So the answer here is experimental design within the living. So in vivo means within the living in Latin. In the computers would be in silico. Test tube or petri dish would be in vitro. Okay. What does the symbol below represent in a pathway? So this symbol. Yep. So an enzyme denotes an enzyme is represented by a diamond shape. All right. What type of relation slash interaction does the following arrow between these two nodes represent? So this line right here, what does this represent? Specifically the arrow. So like this specific arrow like this, what does that mean? Yep. So it denotes an activation relation. Which tool in IPA allows one to keep certain relationships based off set specifications? So specifically keep them. Yep. So the answer is the keep function. So we keep relationships using the keep function, right? What is the measure for risk in case control studies? Yep. So for case control studies, we use odds ratio. That is the statistical method for determining risk. All right. What type of relation slash interaction does the following arrow between these two nodes represent? So specifically this line ending like that, what does that mean? Yep. The answer is inhibition. What do lighter shades of blue denote with molecular activity predictor? So like this lighter shade like this, what does that lighter shade mean? Yep. So the answer is less confidence in predicted inhibition. It's not less confidence in predicted activation because the blue color denotes predicted inhibition. So the answer was this. All right. Which function of IPA allows one to find relationships as intermediary nodes between two or more molecules? Yep. So the answer is Path Explorer. So Path Explorer shows you intermediary nodes slash relationships between two or more molecules, right? Which function of IPA allows one to remove relationships based off set specifications? So remove them. Yep. So use the trim function to take out relationships based off your specifications using the filter options. Okay. Which two studies are considered the highest quality of research? Yep. And as we discussed, systematic reviews and the analysis are both considered at the top of quality and significance of research. What does the shortest path plus one in Path Explorer represent? Yep, and the answer is it shows one additional intermediary node to the shortest paths. Shortest path is the least number of intermediary nodes. So shortest pathless one is just one on top of that. What is a reason for filtering searches in Path Explorer? Yep, and like we discussed, oftentimes our research, that being systematic reviews or meta-analysis, may have certain inclusion and exclusion criteria. So we can use the filter options to include or exclude our criteria based off our needs for our study, right? So this is the answer. Which type of study is often used to determine prevalence of disease? Yep, and the answer is cross-sectional. Like we said, cross-sectional is sort of showing you a picture of the present. Case reports is sort of cases that have already happened. And cohort studies are you're looking at who becomes cases, right? So cross-sectional is because you're looking at a snapshot of the present. It gives you an idea of the prevalence of that certain disease. If buprenorphine were to affect the activity of a molecule after it in a pathway, that molecule would be, so after it comes after buprenorphine. And the answer is downstream to buprenorphine. So if it comes after it in the pathway, it would be downstream. If it comes before buprenorphine, it would be upstream to buprenorphine, right? If buprenorphine were to be affected by the activity of a molecule before it in a pathway, that molecule would be. So should be obvious now. And the answer is upstream. If molecule activity in molecule activity predictor, if a gray line connects two nodes, what does that mean? So this gray line right here, what does that mean in molecule activity predictor? Yep, and it means no predicted effect. So if it's gray, it means that there's no predicted effect in molecule activity predictor. In molecule activity predictor, if a blue line connects two nodes, what does that mean? So a blue line like this, what does that mean? Yep, so the answer is it leads to inhibition. So by decreasing the measurements of morphine, that pathway will lead to an inhibition. So that's basically what that means, that blue line. What is the measure for risk in cohort studies? Yep, so it is relative risk. For case control studies, use odds ratio, but cohort is relative risk. What does the symbol below denote in a pathway? So the symbol right here. So a octagon would denote a function, okay? Which of these is at the bottom of the research hierarchy? Yep, and the correct answer is editorial/expert opinions. Since they do not rely on any empirical data, they are at the bottom of the research hierarchy. All these have some sort of empirical data. What does a dotted line connecting two nodes represent? Like this. Yep, and it is an indirect relationship if it is a dotted line. Which statistical value is increased with the use of meta-analysis? Yep, so power is increased with the use of meta-analysis. If you remember, we discussed that the power goes up with meta-analysis. What does a red molecule slash node and molecule activity predictor represent? So this is red molecule right here, morphine. What does that mean? Yep, so the red color denotes increased measurements. What does an orange line connecting two nodes mean? So this orange line right here, what does that mean? Yep, so it means it leads to an activation. What might be a reason for specifying results in PATH Explorer by its date of publication? Yep, and the answer is research may require most recent literature, like we said, systematic reviews or meta-analysis. You might want the most recent literature included in it. So that might be a reason for sorting by the date of publication. What can be used to reduce the time taken in stage 1 and 2 of the drug design process? Yep, and the answer is in silico methods. If you remember from the presentation, I said that stage 1 and 2, which normally might take years to complete, can be done in weeks using in silico methods. Where would you need to click on to find the grow function? So it will show you a picture of IPA, and this, where would you click to find the grow function? If you can't read it, I have also included over here. what it says. Yep. So to find the grow function, you click on build. So the answer is B. Where do you need to click on to find molecule activity predictor? So if you can't read it, it's written over here. Okay. So to find molecule activity predictor, you would click on overlay. Where do you need to click to create a new My Pathway? And the answer is create new. So the top left-hand corner, there is a create new button. That's where you click to create a new My Pathway. Where would you need to click to find the trim function? The answer is B. So it is also found in the build function, much like the grow function, the path explorer function, and the keep function. Where would you need to click to create a hierarchical layout to your pathway? So you want to find the different layout options. Where would you click? Yep. And the answer was D. You would have to click on the icon labeled by D. As part of the experimental model, what is typically conducted last? Yep, and the answer is clinical trials. In vitro, animal, and in silico analysis is done prior to clinical trials. What relations does shortest paths in Path Explorer include? Yep, and the answer is relations with the least number of intermediary nodes. That is shortest paths. All right. So these are our points. I will be adding up the points from the quiz as well. So just bear with me. I'm just going to be tallying up the points. Okay, so just bear with me. I will be adding up the points from the quiz as well to determine who came in first, second, third, fourth, and fifth. So just bear with me for a little while as I do that. Okay. I'll just go over all of the individual ones right now. Oh yeah, sorry. Yeah, you can leave right now. I'll just tally up all of the points, and I will be posting it on the Google Drive as well. So you would have it on the Google Drive, the final points from the quiz, today's quiz, and including the final points from the previous quizzes as well. So whoever came in first, second, third, fourth, fifth, that will all be posted. So that would take a little bit of time. So I will be posting it later on today. And yeah, you guys are all welcome to leave if you have other commitments. I thank you all for joining throughout this workshop. I hope that this was fruitful for you, that you were able to take away from this workshop, and I really appreciate all of you coming to this workshop. I believe so. Everyone will be receiving certificates for participation. Tallying up all of the points today. So later on today, I will be posting all of the results from today's quiz and the points that were accumulated from the previous quizzes as well. So all that will be posted later today. If anyone has any questions, they are more than welcome to ask anything. Otherwise, that is the end of our session for today. The results will be posted on the Google Drive later today. But yeah, unless anyone has any questions, that's the end of our session.
Dr. Asghar, thanks for running and moderating an excellent session of meta-analysis course for us. Thank you very much once again.